We're in Chapter 7, and this video lesson will cover Section 7.1 and 7.2. Our title says Membrane Structure and Function, so we're going to talk about cell membranes. It says, how does the plasma membrane, so plasma membrane, if you hear that term, it's interchangeable with the cell membrane, but how does the plasma membrane regulate inbound and outbound traffic? So what we're talking about is molecules needing to move across the cell membrane, either to come into the cell or to leave the cell. It says some small molecules move across the cell membrane using passive transport, and with passive transport, we're not going to have to use energy. It says it may require transport proteins, so we'll talk about a specific type of passive transport that requires proteins. It says some molecules use what we call active transport, and those are going to require energy and a transport protein in order to do that. Large molecules will have to move in, so that's the key word there. They're large. We'll have to move in and out using bulk transport, and we've got two types of bulk transport: endo and exocytosis, and both of those will require energy from the cell also. Okay, so let's just kind of look at an overview of these types of transport again. So remember, we are talking about the plasma membrane, so we're talking about that phospholipid bilayer that surrounds every single cell in your body. We can also just call it the cell membrane. So we are constantly needing to get molecules across the cell membrane back and forth, either into or out of the cell. We said that passive transport, you have to be very small molecules can do that, does not require energy, may use a transport protein. Okay, here's an example of a transport protein right here, and we see that our number of molecules is higher on the outside of the cell. We've got a lower number on the inside of the cell, and so those molecules are moving to the inside of the cell, and we'll talk about a few terms that would go along with that process in a few minutes. Okay, with active transport, we said that a cell could move small molecules this way, but they would have to use the transport protein, and we're going to talk about the sodium potassium pump. That will be an example there of active transport where energy from the cell will be required for that process to take place. Now, sometimes we have to move large molecules, and we will call that bulk transport. If we're moving those molecules into the cell, we call that endocytosis. Large molecules are taken in. The molecule will actually fuse with the plasma membrane. The plasma membrane will start to pinch inward right here. Those molecules will enter, and then it will close up right across here. And then the vesicle, which is kind of like the shipping container, will pull those molecules or move those molecules around in the cell. Same thing will happen if we need to get large molecules out of the cell. And so for this example, we could pretend right here in this vesicle that those are proteins that were built at a ribosome. And we could say that if we needed to get those proteins out of the cell, they could be packaged in a vesicle at the Golgi apparatus. That vesicle will then fuse to the cell membrane and become part of the cell membrane right here. Become part of the cell membrane, and then the contents would be expelled outside of the cell. Now, this is going to require energy from the cell. Anytime you're having to fuse with the plasma membrane, we're having to put in our energy molecule called ATP in order for that to happen. And so those will require energy, endo and exocytosis. Okay, so if you wanted to make yourself a little chart so far, we've got passive transport. We're going to talk about three types of passive transport. We're going to talk about diffusion, osmosis, and facilitated diffusion, and we'll come back to this a little later. And then we're going to talk about active transport. We'll come back and talk about the sodium potassium pump, endocytosis, and exocytosis, and we'll talk about those in greater detail. But first, let's talk about the membrane itself. And so section 7.1 says that cellular membranes are fluid mosaics of lipids and proteins. And so that term there, fluid mosaics, means that they are made up of a lot of different types of components that have come together, and then it's fluid, which means it has fluidity to it. It can move around. Okay, so our main components, we're going to have some lipids and we're going to have some proteins. That's the main things, but we're also going to throw in some carbohydrates in there. And so back when we were talking about macromolecules, I explained to you it was important to eat certain good fats, certain good lipids, and then we have to have proteins in our diet and even carbohydrates. And so all of those things come together to build our cell membranes. And so they're mainly composed of phospholipids, and we looked at the structure of a phospholipid in our chapter, in our last chapter. And we said that they have those phosphate heads and those lipid tails. And we said that the heads would have a positive charge and those tails would have a negative charge. And so we call that being amphipathic. And so when they are amphipathic, that means the molecules contain the hydrophobic, which is the water-fearing section, that's their tails, and then they have that hydrophilic, which is the water-loving section. So make sure you understand those two terms, hydrophobic and hydrophilic. Know which part of the phospholipid is which, okay? And so phospholipids will form that bilayer. Remember the bilayer, bi means two, so it's a double layer of those hydrophobic tails that are pointing inside, and then the hydrophilic heads will be exposed to the water side or pointing outwards. It says most membrane proteins, so the proteins are also amphipathic, and so they're going to also have a charge. And so let's look at our structure here. We've got the hydrophilic regions of the proteins will be oriented toward the cytosol. Another word for cytosol is the cytoplasm, so you could also say cytoplasm there, and the extracellular fluid inside and outside of the membrane. It says the hydrophobic regions are embedded in the bilayer. So here's what this means. Here is the hydrophilic heads. Remember, the heads all get a positive, so we've got positive charges all pointing either outwards away from the cell, or we've got those positives pointing inwards toward the inside of the cell. And then the hydrophobic tails, they are all pointing inwards, and it gives this negative hydrophobic region here inside of the bilayer. And because of that, certain molecules will have to pass through this membrane rather quickly to get through to the other side. They're not going to hang out inside of that hydrophobic region. And remember, we said that those tails could have these bends or kinks in them, and that's going to help the membrane to remain fluid. We'll talk about that again in a few minutes. We say that it is a fluid mosaic model, and that's because it's going to be a mosaic. A mosaic means made up of many different parts. It's going to have those proteins bobbing around in a fluid bilayer of phospholipids. So lots of different types of structures all come together here. And so if we look at this picture, we can see that we've got some carbohydrates that are involved here, making up those little tails that are out here on the what we call the extracellular matrix of the cell membrane. Inside we see embedded are some phospholipids and some cholesterol. And those phospholipids and the cholesterol all help to make this remain fluid. And so because it is fluid, they can move back and forth and side to side, and it gives flexibility to our cells. And so we also see some proteins. There's a glycoprotein, which is a combination there of a carbohydrate tail attached to a protein that's embedded there in the membrane of the cell. Okay? And so fluid mosaic model means that we've got lots of different structures put together. It's fluid because of that cholesterol and those phospholipid tails. It says the membranes are held together by weak hydrophobic interactions, which means that it can make and break bonds really quickly, and so it can move around. If the cell membrane needs to, some of those phospholipids need to move side to side, then they can break a bond. Other molecules can come through, and then they can reform that bond. It says most of the lipids and some proteins can move sideways. So we know they can move sideways within the membrane. It will allow certain molecules to pass through freely. And those lipids may even flip-flop across the membrane if they break a bond and something stronger comes along and pulls them across the membrane. As temperatures cool, the membranes switch from a fluid state to a solid state, and so the cell membranes work better at warmer temperatures. And it says the temperature at which a membrane solidifies depends on the type of cells. Okay, so know that temperature is involved there. It says membranes rich in unsaturated fatty acids. Remember, we said that the unsaturated fats are those fats that are healthier for us. They will remain liquid at room temp. They are more fluid than those rich in saturated fats. And so let's look at the difference here and what our cell membranes would look like if we used unsaturated tails versus saturated tails. Okay, and so with the unsaturated tails, remember a phospholipid has these phosphate heads up here on the top, and then they've got I'll just draw a little one out here. They've got two tails, two fatty acid tails, and if those tails have double bonds in the carbons and the hydrocarbons that belong to the tails, then the tails will be kinked, like this example right here. And when the tails kink, it allows for some flexibility in the cell membrane. The phospholipids can't pack tightly, and then it helps to keep it fluid. And so certain molecules can easily pass through the cell membrane if they're small enough, without having to use energy from the cell. If our cell membranes, if they use saturated tails instead of unsaturated, they would look like this over here, okay? And very few molecules would be able to fit or pass through the cell membrane. So they would not be able to fit through, okay? So phospholipids, their hydrocarbon tails, are mostly made up of unsaturated fatty acids. That helps with fluidity. We can also embed cholesterol. So cholesterol can be used in the cell membrane. It says it's a membrane component in animal cells, so we find this in animal cells. It has variable effects on membrane fluidity at different temperatures. And so it will help at warm temperatures. It says at warm temperatures, cholesterol restrains movement of phospholipids. So if they get really, really hot, it will help those phospholipids to stay together. In cool temperatures, it helps those phospholipids to keep that fluidity, keeps them from packing too tightly. And so cholesterol in our picture right here is kind of added into the membrane to help with fluidity so that molecules can freely pass through there. Okay? It says membranes must be fluid to work properly. Fluidity affects both permeability and movement of transport proteins. And so membranes that are too fluid cannot support protein function. We want them to be fluid, but not too fluid, because we still have to produce proteins within the cell. If it's too fluid, then it shuts some of that down. It says organisms living in extreme temperatures have adaptive differences. So if they live in extreme temperatures, we're going to see that there is a difference in their lipid composition. One example of that is in the tails, the hydrocarbon tails in fish of their cell membranes. The hydrocarbon tails, I guess I need to erase that, start again there. The hydrocarbon tails in fish that live in really, really extremely cold environments will have a high portion of those unsaturated fats so that their cell membranes can remain fluid because of those cold temperatures that they live in. It says, for example, winter wheat, which is a plant, has the percentage of unsaturated phospholipids increases in autumn. So they kind of prepare for winter coming. So they're going to increase those unsaturated fatty acid tails and phospholipids in their membranes, and it prevents solidification during the winter, and so they're able to survive winter that way. Okay. It says a membrane is a collage of different proteins, and so we've been saying that it was a mosaic, and this is an example here of a tile mosaic where they have used many different sizes and shapes and colors of tiles to produce this piece of artwork. Your cell membranes are kind of like that. They're a piece of artwork. They're made up of many different types of proteins, many different lipids, many different carbohydrates, clustered in groups, it says, embedded in the fluid matrix. And so phospholipids, remember, are the main component, but then we've got those proteins, and those proteins determine the most of the membrane's functions. And so those proteins tell the membrane what to do and which molecules to allow in, which molecules to send out. So those proteins are kind of running the show. It says the protein composition of membranes varies among cells within an organism. So different types of cells, so blood cells will have different types of proteins on them than a blood cell would. A blood cell will have different types of proteins than a bone cell would. Okay, there are two major types of proteins that we're going to find that will be part of the cell membrane. We've got the peripheral proteins, and those will be bound to the surface. And so here they are. Here's an example of a peripheral protein. Just kind of attached there to the surface. Here's some more examples up here pointing to these two that are just attached there to the surface. And then we've got integral proteins. Integral proteins run throughout the cell surface, so they penetrate the hydrophobic core. And so here's our integral protein right here. And these integral proteins we're going to see are important for moving molecules in and out of the cell. And then the peripheral proteins are going to be important for cell signaling, cell identification, and then sometimes we might see that coenzymes will attach to those types of proteins to allow a certain enzyme to work. And so both have very important functions within the cell membrane. So know that there's two types: peripheral, just found on the surface, bound to the surface, and then integral. They're going to run all the way through from one side to the other. All right, the cell surface membrane proteins, so both types, peripheral and integral, carry out many different functions. For transport, we may be transporting materials through the cell membrane using the help of those transport proteins, both passive transport, so this would be an example of passive transport, and active transport where we're trying to use ATP to move things. We'll have to use the help of proteins to do that. Enzymatic activity, we know that enzymes are proteins. Those can be attached to the cells. Cell signaling or cell transduction right here would be an example. Those proteins, a signaling molecule will attach to the protein and direct some type of cell activity inside the cell, so we have proteins for that. Cell-to-cell recognition, so a blood cell can recognize another blood cell based off of the proteins and the carbohydrate tails that are attached to the surface of the cell. Also, intercellular joining. Sometimes cells come together to form tissue, and they attach themselves together using those proteins that are found on the surface of their cells. And then they can also attach the cytoskeleton, the extracellular matrix can attach itself to the actual cell membrane using those proteins. And so lots of functions there for proteins. If you are looking at this in PowerPoint slide, that would be an active slide right here. You should be able to click that and see an animation of the functions of the plasma membrane. So watch that if you're able to watch that. It says the role of membrane carbohydrates in cell-to-cell recognition. So what do we have carbohydrates for in the cell membrane? It says cells recognize each other, so they're going to use that to recognize each other by binding to molecules on the surface of the membrane. It says many of these surface molecules are bound to short branched chains of carbohydrates. So we're going to have two main types. We've got glycolipids and glycoproteins. And so glyco kind of sounds like glucose, which tells us they are carbohydrates. And glycolipids are carbohydrates that are bound to lipids. And then glycoproteins are going to be carbohydrates that will bond to proteins. And both of those are very important for cells recognizing each other. This is one reason why you have to be given a very specific type of blood if you needed a blood transfusion, is because of those proteins that are found on the outside of your blood cells. They would not want to mix together. You got to have that same protein on the outside of your blood cells if you had to have blood given to you. It says the diversity of surface carbohydrates enables them to function as markers. So those carbohydrates will be markers for cell identification. And then the membranes have a distinct inside and outside face. And so those carbohydrates will mark the inside and the outside of the cell, and then also within organelles that are found inside the cell, like the membrane around the nucleus, or the membrane around the Golgi apparatus, or the membrane around the mitochondria. All of those membranes will also have those carbohydrate markers on them. And so here's an example of that. Let's just use the Golgi apparatus. So here's the Golgi apparatus, okay, pointing right here. And we can see that it's got some glycolipids that are attached to it, and those little tails, those carbohydrate tail portions that are attached are showing the sidedness of the Golgi apparatus. So it's reminding these molecules that are coming into the Golgi apparatus or leaving the Golgi apparatus which side it is on, okay? And so they have a very important function as far as keeping things straight as far as which side is which. Here are some more. You can see these little membrane glycolipids, these little green molecules that are attached all along the outside of the cell membrane. And that is letting other molecules and other cells know what type of cell this is and what side of the cell membrane this is. Okay? All right, we're going to end this video lesson. Well, I guess we'll keep going for section 7.2 and then we'll end it. Okay, so it says membrane structure results in selective permeability. That word's hard to say. It says the plasma membrane controls the exchange of materials between the cell and its surrounding. So that's its function. So that's the main function. We'll control the exchange of materials. But in order to do that, some things are going to have to cross the membrane. And so these membranes are selectively permeable, or sometimes we may say semi-permeable. You may hear that term also, which means some substances cross more easily than others. And so some have to work a little harder than others to get across the membrane. We're going to see that sometimes that will require energy from the cell, and then sometimes that can happen for free of charge to get across. It says the fluid mosaic model explains how membranes regulate molecular traffic across the membrane. And so this is just a little generic picture here. I know it looks blurry, but I'm not really wanting you to label anything, but just understand that we've got this fluid mosaic model here, all of these different carbohydrates and proteins and lipids all working together so that things can cross this membrane. We want to effectively get things in and out of the cell. It says the permeability of the lipid bilayer. We've got the hydrophobic, so remember hydrophobic fears water, and those are going to be nonpolar molecules, will dissolve in the lipid bilayer and pass through the membrane rapidly. Hydrocarbons would be an example of that. Carbon dioxide can do that. Oxygen can do that. It's all free of charge. Okay, not going to cost any kind of energy. Now, it says the hydrophobic interior of the membrane impedes the passage of hydrophilic and they're going to be polar. So remember, hydrophilic polar molecules are going to have that positive or negative charge. Examples would be sugar or water or ions. And we're going to see that we'll have to have a special way to get these type of molecules across, which is going to require some type of protein. Okay, some of these we can still move across free, so we'll still call that using passive transport, but some of those are going to use energy, so they will have to use active transport. Okay, so as long as it is non-polar, doesn't have a charge, and it is able to dissolve into the membrane, so hydrocarbons, which means molecules made up of carbon and hydrogen, carbon dioxide and oxygen, can just move through the membrane. These other ones, sugars like glucose, water, and ions like sodium and potassium, will all have to move with the help of a protein to get through this phospholipid bilayer. Okay, remember we have the hydrophobic, which is that negatively charged region inside, and then we have the positively charged phosphate heads on the outside. So hydrophilic would be on the outside, hydrophobic on the inside. All right, so I keep saying we have to have the help of some proteins to get certain things across the cell membrane. It says hydrophilic substances cross the membrane more quickly, and they're going to pass through transport proteins in order to do that. So if you're hydrophilic, you cannot dissolve into the membrane, and we're going to have some help to get across. It says channel proteins will have a hydrophilic channel that certain molecules or ions can use as a tunnel. So here we go with the channel protein in the picture. It's just like a passageway, it's like a tunnel. They can move straight across the tunnel, either to get in or out of the cell. Carrier proteins are similar, but it says they're actually going to change shape in order to shuttle these molecules across their membrane. And so if we look at the picture here, we've got the carrier protein, it's in green. The molecule we're needing to get across is here. It will move into the channel, or into the carrier protein, I'm sorry. The carrier protein will change shape, and then it will allow that molecule to move through. This is active transport, which means it's going to require ATP, which is energy. Okay, so we've got two types of proteins that move things with passive transport, no energy needed. We're going to use channel proteins. With active transport, we're going to use carrier proteins. That's going to require energy. Okay, now one channel protein that we use is called, so this is a channel protein, they're called aquaporins. Aquaporins are going to increase the rate of passage of water molecules. So that's how we get water molecules into the cell. They will pass through aquaporins. And it says overall up to 3 billion water molecules pass through the cell membrane per second. And so our cell membranes are very, very busy moving these water molecules across. And this is why you become very ill very quickly if you become dehydrated, because we need a lot of water to perform all of the chemical reactions that are taking place inside of our cells. Okay, if you're looking at this on PowerPoint slide, this video should be active. I think let me see if it'll play. I don't think it's going to play right now with the one I'm on, but you should be able to click that video there and see those little molecules moving through the aquaporin. It's just an animation there of that molecule. Okay, so aquaporins will move water. That is still going to be passive transport, so it's not going to require energy. It will be free for the cell to do that, but it's going to have to move those water molecules through an aquaporin because water molecules are polar. They have a charge. Okay, all right, transport proteins move only very specific substances. And so, example, we have a glucose carrier protein. Guess what it transports? Only glucose. They will not transport even fructose or any other type of sugar. It's only for glucose. And so it says the selectively permeability of a membrane is dependent on both the lipid bilayer and the specific transport proteins it contains. So it's got to get through the lipid bilayer, and it's also got to get through those transport proteins. And certain proteins transport certain molecules across the membrane. Okay, so a glucose carrier protein would transport glucose. An aquaporin would transport water. So they're very, very specific. Okay. All right, we're going to end section 7.2 there. We're going to pick up in the next video lesson with section 7.3. And we're going to talk about passive and active transport here coming up. We're going to list several types of each one of those.